Our next presentation is Metagenomi, and I'd like to welcome Jean Irish, President, Chief Operating Officer, Executive Board Director, to the stage.
Great, thank you for the introduction. Good morning, everybody. As we're shining a spotlight next door on the gene editing platform approach to improve development efficiency, I'm very pleased to share with you here the progress we have made at Metagenomi, a precision genetic medicines company, where we apply our proprietary next-generation gene editing platform to develop curative therapeutics. Before we proceed, please note this presentation contains forward-looking statement and ask that you refer to our SEC filing. Gene editing holds the promise of curative therapeutics, while first-generation gene editing companies made groundbreaking progress, including first FDA-approved gene editing therapy and multiple programs in clinical development now. Many diseases still cannot be addressed by current existing technologies. A new wave of next-generation technology is needed to address the full spectrum of genetic diseases. Our AI-driven metagenomics platform is at the forefront of the next generation of gene editing technologies. We are harnessing the power of metagenomics, unlocking 4 billion years of microbial evolution on the planet to discover and develop a suite of programmable gene editing tools with capabilities to make precise human gene modifications and to translate these tools into curative genetic medicines. Metagenomi was founded based upon the pioneering science of metagenomics. By studying the samples collected from diverse microbe-rich ecosystems using artificial intelligence, ancestry state reconstruction, proprietary algorithms, and high-throughput screening to reveal novel editing machinery, we have identified over 20,000 novel gene editing systems to date. This process results in our proprietary toolbox where we're nearing the capability of correcting any genetic mutation anywhere in the human genome. This slide illustrates our expansive collection of gene editing tools. On the left hand of the slide, you can see our programmable nucleases that form the backbone of our toolbox. These nucleases can knock genes in or knock them out with diverse targeting abilities. Some of them are ultra small, that are overcoming several major limitations of first-generation CRISPR-Cas9 systems. Because of our extensive experience with these programmable nucleases, we can think of them as chassis upon which we can build our tools. For example, with deaminases from our metagenomics database, we built the base editors that represent some of the smallest and potentially most efficient systems in the industry. The same goes for RNA-mediated integration systems, we call RIGs, formed with novel reverse transcriptase. Our Cas are at the very forefront of the next generation of gene editing systems to address the challenge of a large, targeted genomic integration using DNA. Using high-throughput testing, we validated thousands of novel CRISPR nucleases. Here we highlighted the high editing efficiency of a sample of several leading systems. A key point here is that this amazing level of efficiency is natural. We have no need to resort to any protein engineering to achieve these levels of activity. Each of these nucleases has specific targeting profile. Having a large collection allows us to select the ideal nucleus for any given gene editing in design genetic medicines. Metagenomi smart nucleases are distinguished by their small size, around 450 to 1,000 amino acids, in contrast to the substantially large SP-Cas9. Given the small sizes, these smart systems are within the packaging limits of AAV, which is very important. As today's lipid nanoparticle delivery technology for gene editing has limitations in access disease targets in organs other than the liver. These smart systems have the potential to address diseases such as in CNS, neuromuscular, or heart, to both as a nucleus and a base editor for all-in-one AAV delivery. We have published smart nucleus in the past and will highlight the development of these smart base editors at the upcoming international conference on CRISPR technologies in San Diego next week. Base editors are powerful gene editing tools that are leveraged to knock out the genes through single base transitions without DNA double-strand break. Our best-in-class adenine base editor meets all critical criteria necessary to achieve successful multi-target gene editing with remarkable targetability, efficiency, specificity, and tolerability. We will present additional data at the ESGCT later this month, but here I would like to highlight a few key points with you. Our base editors are highly active with significant wider targetability than first-generation base editors. As an example, our adenine base editor allows us to target over 95% of base pairs in the human genome. This is 5 times more than typical SP-Cas9 base editors. Our adenine base editor consistently achieves greater than 90% editing efficiency across multiple gene targets in primary T-cell engineering. Specificity is very important for gene editing. No genome-wide deamination, no detectable translocations has been observed with our base editor. Finally, great tolerability in cells was evidenced by cell health readouts, no impact on viability, expansion, and gene expression post-editing in our multiplex editing of primary T-cell. For large targeted genomic integration, first, our dual-vect approach by delivering the gene editing cargo in lipid nanoparticle and the DNA template in AAV has demonstrated therapeutically relevant durable protein expression in NHP. I will share more data with you in this presentation. In addition, the big rigs represent Metagenomi's second RNA-mediated integration pillar. We present what we believe to be the first ever targeted integration of greater than 900 base pairs in human cells with all RNA delivery. Furthermore, metagenomic Cas are a new technology that allow programmable site-specific integration of tens of thousands of base pairs. Here, demonstrated is our Cas system capable of targeted DNA integration into the genome of human cells. We apply this diverse toolbox to unlock the development of curative medicine, so we focused on indications with clinical validation and where we have strong conviction in the underlying biology, where there is a readily available biomarker that allow us to achieve rapid clinical proof of a concept. Where there is an established development pathway and where gene editing can make an important advance in the standard of care for patients. Our strategy is to focus initially in the liver indications where we can move forward quickly with tools that are available today coupled with available delivery systems, and where the regulatory landscape is becoming increasingly clear. We then build on those experiences with both more complex gene editing tools and more complex delivery systems. Turning to our specific pipeline based on this strategy, our most advanced program is in hemophilia A. We recently declared our development candidate. Additionally, we see an opportunity to leverage the platform to rapidly advance additional therapeutic candidates targeting secreted protein disorders. We are also working with our partner Ionis on 4 cardiometabolic programs. We believe the rapid progress we made together in this collaboration is evidence of our ability to execute our strategy of translating novel gene editing systems. We're leveraging the valuable insights gained from our leading programs to progress to more complex gene editing with our advanced technology. as exemplified by our programs in A1AT and Wilson's diseases. While the majority of our efforts are focused on in vivo genetic medicines, we offer our technologies to partners in the cell therapy space to enable advanced cell engineering that would require multiplex knockouts and non-viral knock-in of the transgenes. We recently achieved an important development milestone with our partner Affinity, where we hold drug master files for providing them with GMP gene editing reagents and regulatory filings. Finally, we plan to utilize our ultra-small editors delivered by AAV to address compelling neuromuscular diseases and seek to maintain our leadership in the area of specific large gene integration using RNA and DNA-mediated systems for diseases including cystic fibrosis. Our lead program aims to address the need for hemophilia A, that is the most common X-linked inherited bleeding disorder caused by mutations in factor VIII gene leading to loss of functional factor VIII protein. There are nearly 30,000 patients in the US, more than 500,000 globally. About 60% of those who have severe diseases are at the greatest risk of spontaneous life-threatening bleeding events, as well as progressive joint damage. Despite improved outcomes, the standard of care incurs a high treatment burden on patients, involving lifelong repeated IV infusion of recombinant factor VIII on patients, recombinant factor VIII, or injections of an engineered factor VIII bypassing agent, resulting in high economic burden with estimated lifetime cost of $15 to $18 million per patient. Rather than provide the factor VIII gene in an abnormal location outside of a genome, such as done in gene therapy, which risks dilution over time from cell division and epigenetic silencing from cellular machinery, our approach is to insert a factor VIII DNA cassette directly into safe harbor location within the genome. Our approach would allow us to address pediatric patients who have the most to benefit from a single potential curative treatment that provides lifelong bleeding protection. Shown here is the result from our recent study, the 12-month durability data from non-human primates. The results are truly groundbreaking, representing the first site-specific large gene integration of the factor VIII gene, resulting in therapeutic level of expression and representing a functional cure in these animals. We have observed sustained factor VIII activity through the time point in all three animals, well within the durable therapeutic range of approximately 10 to 150% of normal. We also succeeded in obtaining this expression with an integration rate of only 1 to 2%, so that not to disrupt the albumin production. Importantly, our study has revealed no safety signals of concern, and no off-target editing has been identified to date. The progress we have made on this program not only validates the efficiency and specificity of our novel nucleus in non-human primate models, but it also serves as a valuable platform to address other diseases characterized by deficiencies in secreted proteins. It's a true platform approach. We believe our ability to develop and characterize complex human genome editing components is essential to pursue a successful regulatory pathway for genetic medicines development. Along with our pipeline, we strategically invest in automation, RNA optimization, state-of-the-art characterization assays, CMC development, and GMP manufacturing capability with the in-house and CMO network for our own programs and many of our collaborations. We set out to build a premier gene editing company. Overall, this has been an exciting year so far for Metagenomi, showcase our ability to rapidly translate our technology innovations into pipeline programs. Based on our highly efficient, precise, and diverse gene editing toolbox underlined by wholly owned intellectual property, we